All right, welcome back to part 5 of Chromosome Organization. So just as a reminder of where we were, we have DNA. DNA wraps about twice around these histone octamers made up of 2H3, 2H4, 2H2A, 2H2B, creates a hockey puck type of structure. DNA wraps nearly twice around it, and it creates this bead on a string type of conformation that is further compacted with the H1 protein um, that helps link the linker regions together. And then it condenses into these 30 nanometer fibers, again, the structure of which we are a little bit unclear. But both of those two events has shortened the DNA by about 50-fold. Right? The third level of compaction involves the interaction of this 30 nanometer fiber with the nuclear matrix. All right, so what is the nuclear matrix? If you can remember back in Bio 101 or your biology classes, we have a nucleus. The nucleus has a membrane wrapped around it. Directly underneath that membrane is something called the nuclear lamina. And this is made up of fibers. Again, these are cytoskeleton fibers, intermediate filaments specifically, a specific type of intermediate filament that lines that internuclear membrane and it gives it structure. Remember, the membrane itself is nothing more than a piece of fat, or it's a very oily in nature. So we have the nuclear lamina, but there are also internal matrix proteins. So some of these fibers are connected to the nuclear lamina itself, but it fills the entire interior of the nucleus as well, kind of creating a gel-like material. So the inside of the nucleus is very gel-like because it has all of these fibers running through it, all these filamentous matrix proteins. So here's a schematic. So here we have the nuclear membrane. Inside of the nuclear membrane, we have these pores that go through. Those pores are where messenger RNAs are going to come out. We have the nuclear lamina, which lines the inside of it, but we also have these fibers that run through and proteins bound to it. Okay, so keep in mind that we have all these filaments running through. What happens is we take this 30 nanometer fiber, and remember, this is not referring to a strand of DNA. This is referring to this. So we have one of those, and it is attached by matrix attachment regions in the DNA, which bind to these proteins that link it to this nuclear matrix. Okay, but it leaves these loops extending out away from that filament. So we have a bunch of these, what we call radial loops, and these can extend up the 25,000 to 200,000 base pairs. And these loops are typically where you will see genes being present. The regions down here where we see the 30 nanometer fiber connecting to the nuclear filament is generally where we see some of those repetitive sequences. All right, now this attachment has two major roles. One is it further compacts the DNA because now instead of having just one long loop, we have a loop, or one long strand, we have it condensed into a loop, this radial loop. The second thing that it does is it also serves to organize the chromosome within the nucleus. So oftentimes I might refer to the nucleus as kind of a bowl of spaghetti. And that serves well for an analogy when you're speaking about the steps of mitosis, about it condensing down. But what I want you to understand is it's actually much more organized. So here we have a picture of a cell, and it's showing a cell in two different phases. So here is the cell during interphase, and here's the cell during metaphase, where we have all the chromosomes compacted down into that metaphase chromosome. What we've done is stained each chromosome a different color. So we've stained chromosome 1 red, we've stained chromosome 2 green, and so on and so forth. If we do the same staining procedure on an interphase cell, this is what it looks like. So you'll notice that chromosome 1 just isn't randomly scattered about, it's organized in a region. And chromosome 2, chromosome 3, we see the same type of thing. There's definitely a lot of organization. And so that is the second thing that this radial loop structure achieves. The fact that this is kind of uniform through the interior of the nucleus, and we have all these loops, we can achieve this structure. Now, when we look at the DNA present in an interphase cell, we will see some additional condensation or some additional compaction that are specific in certain regions within a chromosome, and other regions that don't have that compaction. So when we see DNA that is even further compacted from that radial loop type of structure, we'll refer to that as heterochromatin. Okay? When it is less condensed, more in that just straightforward radial loop, we'll call that euchromatin. Now, an important distinction here is that heterochromatin is transcriptionally inactive. So what cells have done is taken pieces of DNA that has genes that it doesn't care about or doesn't need, such as muscle fibers in a brain cell. Those genes aren't needed, so that region of DNA has been bound up more tightly to keep it out of the way. And also by binding it up tightly, it makes it harder to accidentally activate some of those genes. So in that, we have the radial loops, but they're even further compacted, and I'll show you how that occurs. The euchromatin is transcriptionally active, and it is made up of those 30 nanometer fibers in radial loops. And those radial loops can be transiently unwrapped to allow transcription to occur. Now, there are a couple different types of heterochromatin. So there is constitutive heterochromatin, and these are regions that are always bound up in heterochromatin. They are permanently inactive to transcription, and they usually contain very highly repetitive sequences. Where we see this constitutive heterochromatin is in regions such as the centromere and in the telomeres. Those are regions that don't have genes. They serve a separate type of purpose, so we can package it up tightly to keep it out of the way. This facultative heterochromatin is regions that might be able to convert in between euchromatin and heterochromatin at different levels. So sometimes it's more permanent, sometimes it is more transient depending on what the cell needs. An example of this is something that we call the bar body formation during female development. So we know that um, biologically male humans have one X chromosome and one Y chromosome, and biological women have two X chromosomes. One of those X chromosomes is packaged up and not used at all. This helps keep a balance of the genes. So since the biologically male cell is adapted to use only one X chromosome, a biologically female cell will package up one of those so the genetic balance is equivalent between the two. All right, we are also going to take a look at how we can take that, how we can start from DNA that is completely unwrapped and how we ultimately can end up with these metaphase chromosomes. And the proteins that carry this out are something called condensin and cohesin. So when cells are getting ready to enter mitosis, they need to take those radial loops and those regions that might be contain heterochromatin, and we need to package it up even tighter, right? So let's kind of progressively go through these different levels of compaction, right? So here is a naked strand of DNA. It gets wrapped around these histones, and we have these H1 linker histones. We get this bead on a string type of conformation. This condenses into something called a 30 nanometer fiber. We're not entirely sure about how it's laid out, but it's something along this line. We take those 30 nanometer fibers, we periodically attach that 30 nanometer fiber to a protein filament, and this is part of the nuclear matrix. What then starts to happen is when we want to take this euchromatin and turn it into heterochromatin, we need to take these scaffold proteins that the DNA is bound to and loop them in on themselves, right? So this is what euchromatin looks like. This is what heterochromatin looks like. If we continue doing this even more, we'll ultimately end up with this metaphase chromosome. All right, so in the metaphase chromosome, all these radial loops are highly compacted and stay anchored together to its scaffold, all right? And the histones are needed for this compaction. So here is a metaphase chromosome, and if we take it and treat it with salt, salt will drive off the histone proteins, and you'll see all that DNA spill out here. So the histones are achieving a bulk of it, and here we can actually see the scaffold is still intact in the background. So it's really the scaffold that's being linked together to create the structure, and it's being in this conformation in a metaphase chromosome is dependent upon those histone proteins already having the DNA wound up. So what achieves this tightened scaffold? Well, it's these two proteins, condensin and cohesin. These are both called structural maintenance of chromosome proteins, or SMC proteins, and they use the energy from ATP to achieve a change. It is essentially a big clamp-like protein, and its action is driven down at this end, where ATP binding and hydrolysis will cause it to open and close. What happens is these cohesin